For this lecture, we will cover Chapter 1, which covers the highlights of innate and adaptive immunity. Louis Pasteur is generally considered to be the father of immunology. Now, immunology itself is the study of molecules, cells, organs, and systems that are responsible for the recognition and disposal of any non-self materials or foreign materials, any sort of invaders. And it studies how they actually work and can be manipulated. So all aspects of the body's defense, such as antigens and antibodies, allergies, and hypersensitivity type reactions, are all included as part of immunology. Historically, we've thought of immunity to refer to the resistance of pathogens based on the ability of the immune system to actually recognize and dispose of those foreign materials or those non-self materials. Today, as we've kind of advanced and learned a little bit more, we know that the immune system plays a critical role in not only just our overall health, but also in various disease processes. So there are some desirable consequences of actually having immunity. So this would include things like having an actual natural resistance, being able to recovery and actually acquire resistance to an infectious disease, as well as having the advantage of us actually being able to scientifically study various aspects of immunity and being able to manipulate the immune system to actually stimulate protection against various diseases, as well as to treat a variety of various clinical conditions. Now, if there is some sort of deficiency or dysfunction within the immune system, then it can actually cause a variety of diseases. So undesirable consequences of actually having immunity can include things like allergies, rejection of organ transplants, and various autoimmune disorders. So with your autoimmune disorders, this is where the body actually thinks that its own tissue is foreign and it will actually start attacking its own self. So that's where the auto part comes in. Now, the overall immune system itself can be divided into two parts. You have innate immunity or natural immunity, native immunity, inborn immunity. And this actually provides your immediate protection component. And then we also have adaptive immunity. And this particular type of immunity is a little bit more, it's slower and has a more defensive, focused defense component to it where it targets something specifically. Now, with innate immunity, it actually reflects the person's ability to resist infections using the first and second lines of defense. With this system, the whole defense process is not really based off of any sort of memory of exposure to whatever it is. It's basically the body's way of kind of protecting itself on a more immediate level. Whereas with adaptive immunity, it's more specific and it exhibits a memory of that prior exposure, and it actually uses that memory for antibody formation as part of that process. Now, we will kind of talk about these individually in a little bit more detail. Now, with the innate and adaptive immune systems, they do both kind of do the same thing as far as defending the body, but they do it different ways. With the innate immune system, it is an ancient form of host defense, and it appeared before the adaptive immune system was really well understood. So it is actually composed of a diverse array of hemopoietic cells. So this is going to include things like the dendritic cells, the monocytes, macrophages, granulocytic lines, natural killer lymphocytes. Innate lymphoid cells have actually been more recently identified as cells that actually have a part in the innate immune system. And so these cells have an emerging role in controlling tissue hemostasis and when there are situations of things like infection, chronic inflammation, metabolic diseases, cancers. The innate immune system and the alternative complement pathway, which we'll discuss a little bit more detail here shortly, are activated immediately after an infection, and they start to actually control manipulation of the infecting organism. Innate immune recognition is mediated by germline encoded receptors, which means that that the specificity for each receptor is genetically predetermined. So germline encoded receptors have evolved by natural selection. So it means that they actually have a defined specificity for infectious microorganisms. The problem is that every organism has a very limited number of genes that it can actually encode for within its genome. But in contrast, the adaptive immune system is organized around two particular cell classes to include the T and the B lymphocytes. Now, when an individual lymphocyte encounters an actual antigen that will bind to its antigen receptor sites, then activation and proliferation of that lymphocyte actually starts to happen. And so this is called clonal selection, and it is actually responsible for the basic properties of the adaptive immune system. Now, the random generation of a highly diverse database of antigen receptors actually allows the adaptive immune system to recognize virtually any particular antigen. However, the downside is that this recognition actually is the inability to distinguish something that is foreign from a self-antigen. So activation of the adaptive immune response can be harmful to the host when the antigens are self or environmental antigens, because this can trigger various processes where these antigens can actually mimic something else, and then it can start triggering some sort of autoimmune response. Bacteria, fungi, protozoa, viruses, all of these things can colonize our skin, eyes, ears, nose, mouth, anus, vagina, and gastrointestinal tract. So the term microbiome actually refers to all of the microbes that inhabit the various locations of our bodies. So this is a relatively new field of science where it has actually been discovered that there are very unique and different mixes of microbes that each individual has. And most related studies actually investigate the relationship of the microbiome to our health and various disease states. Now, the guts or gastrointestinal tract of mammals actually harbors trillions of microorganisms, and we call these microorganisms microbiota. So recent evidence actually suggests that the host microbiota and the immune system interact in order to maintain tissue hemostasis in a healthy individual. Among the numerous microorganisms that you can actually have within the gut, some of the bacteria are actually known to provide health benefits to the host when they are actually acquired or you have them actually in very adequate amounts. So when you have enough of that particular good microorganisms or good bacteria, then this is actually considered to be a probiotic. Now, probiotics and other beneficial bacteria will actually provide colonization resistance to pathogens. So in other words, as long as you have an abundant number of the normal stuff, then it keeps those abnormal pathogens from actually causing any sort of complications or infections. Now, those beneficial microbes can actually indirectly diminish the pathogen colonization, so can actually kind of help with keeping that colonization of those pathogens down by actually stimulating the development of the innate and adaptive immune systems, in addition to the function of the mucosal barriers that we actually have throughout our body or throughout our GI tract. Now, microbiota actually regulate the development and differentiation of local and systemic immune and non-immune components by doing the following. Actually, through the regulation of innate immune functions and hemostasis, the regulation of adaptive immune functions in the intestine, and the regulation of systemic and innate adaptive immune functions. Now, if there is any sort of disruption to the host microbiota, especially within the gut, then it's actually been shown to be associated with various autoimmune diseases. So now let's kind of break down those lines of defense. So with innate immunity, that's your early defense system, and it responds almost immediately to microbes and injured cells. Now, if there's any sort of repeated exposure, it can actually invoke an identical innate response to the groups of related microbes. So as long as they're kind of in the same broad category, then the body's going to have the same response every time. There are 3 lines of defense that exist within the human body. The first 2 of these defenses are the principal components of innate immunity. So if a microorganism actually overwhelms the body's natural resistance and overwhelms those first 2 responses, then you'll have a third line of defense that will actually kick in. And this is your acquired or adaptive immunity, which allows the body to actually recognize, remember, and respond to some sort of specific stimulus or an antigen. Now, the first line of defense is a set of barriers between the internal and external environments. So this is basically like your skin layers. The second line involves the innate inflammatory response. So this is going to include like your phagocytosis. And then the third line includes the adaptive immune response and the innate defense that is actually offered by the natural killer cells. Now, of course, tumor cells that can arise within the body are not affected by these lines of defense. As we talked about within hematology, that most of these cells are due to some kind of defect that has taken place within that cell, some sort of genetic defect. So if that's the case, you're actually going to need that third line of defense to help with those particular cells. So this diagram here, this picture here, it's actually kind of showing those lines of defense where you have a breach in that skin layer, and then you have those bacteria actually enter, and then you have that macrophage that's actually going to do that phagocytosis, and then from there you're going to have those T cells that are going to kind of respond to that particular bacteria so that it can develop its particular response to that bacteria as far as creating antibodies or using any sort of like natural killer cells to actually defend off that bacteria. So you can kind of see where all 3 of those defense lines are in action there. Now, before a pathogen can invade the body, it must overcome the resistance that's already provided by the body in that first layer of defense. So the first barrier to infection is going to be unbroken skin and mucosal membrane surfaces. So these surfaces are actually essential in forming some sort of physical barrier against those microorganisms, because this is where the foreign material is actually going to be first introduced to the host. Now, of course, we know that like with our skin or layers of skin, that the skin's constantly renewing. You have those epithelial cells that are constantly coming off of the skin as new epithelials are made. So they're all going to assist with, that's all going to kind of assist with keeping that new healthy live tissue in there, sloughing off any of that bacteria that may be on there that's not needed. Of course, you have your normal microbiota that's going to help to prevent any sort of pathogen colonization of the skin or any of those mucous membranes. And so all of this together is going to kind of work together to eliminate any of those foreign microorganisms that may be introduced. Now, in addition to the actual physical barriers, you do have the ability to wash away any sort of potential pathogen. Your tears and saliva actually have chemical properties that help defend the body. So they have enzymes called lysosome, which will actually attack and destroy the cell wall, which makes that bacteria a little bit more susceptible. And so from there, IgA antibody, which is formed by the adaptive immune response, helps with being an important protective substance that's actually in the tears and the saliva. You have various secretions throughout the body that are part of that first line of defense to keep from any sort of microbial invasion. So the mucus that's actually adhering to the membranes and the wet mucosal surfaces of the nose, pharyngeal, they actually will trap those microorganisms, and then they can be expelled out through things like coughing, sneezing, blowing the nose, things like that. So that kind of helps with that extra layer to remove that. You also have various oils that are produced through the skin. and that are secreted through the various glands, as well as lactic acid that you can find in sweat. So that has an antimicrobial type of property. Earwax itself can actually protect the canals within your ears. And then you also have secretions, various places throughout the body that kind of help with elimination of liquid and solid waste. So this will be like within your gastrointestinal tract, urinary tract. So they help to remove anything and keep it from actually sticking to the walls of those various tracts. Things like the acidity, how acidic or how alkaline the fluids are that are within like the stomach, intestinal tract, vaginal area, that can all have a factor in whether or not those organisms live in that area. So like within the vaginal tract, you have certain bacteria that like a pH of a particular level versus some that like a pH at another level. So depending on the pH of the vaginal tract, then that can it can kind of help determine that particular environment and what will actually grow and live in that environment. Within like the respiratory tract, you have the cilia that actually help to kind of move things in and out. So your body has lots of defense mechanisms. So as long as everything's working properly, you really shouldn't have too hard of a time getting rid of and eliminating a lot of these things before they ever cause a problem. So once those initial barriers have been breached, then the body can kind of start kicking in that innate or natural inborn immunity. And so what it will actually do is start kind of defending the body once that first line is breached. So acquired resistance, which is specifically recognizing and selectively eliminating exogenous and endogenous agents, are going to be part of this process. Now, the innate immune system predates the adaptive immune system, and it is actually common to all living organisms. So all multicellular organisms have the same challenges. We all have to deal with bacteria, viruses, parasites, fungi. So natural immunity itself is a very non-specific mechanism. And so the unique characteristics of the innate immunity actually include rapid recognition of microbes. You don't have to have any sort of prior exposure in order for it to work. Uses widely expressed non-variant receptors to recognize microbes, and receptors that will distinguish between non-self and self. Now, if you actually have the microorganism that passes that skin and mucosal membranes, then the second line of defense will kick in. Now, it will also involve a cellular and a humoral defense mechanism that will actually start to be initiated. So with our humoral mediated immunity, it will actually involve complement lysosomes and interferon, whereas your cell mediated, it's going to involve your mast cells, your neutrophils, and your macrophages. Now, in order to kind of understand how that second line of defense works, I just want to kind of add in that different classes of viruses, they're kind of divided out a little differently. You've got the double-stranded RNA, DNA, unkept RNA. So depending on the particular virus that someone is exposed to, the body will actually develop pattern recognition, or the body actually has pattern recognition receptors, which will actually determine if whatever molecule it's being exposed to is actually immunogenic. So those pattern recognition receptors will actually detect specific viral components or viral intermediate products, and then they will induce some sort of pro-inflammatory cytokine type of response in those infected cells or any of those cells that are part of that immune process. Now, you can actually have tissue damage that results from this process due to biochemical and cellular changes that happen in that area. So that's something that we do have to definitely be very mindful of. Now, complement proteins are the major humoral or fluid component of natural immunity. So there are other substances of humoral component that include lysosomes and interferon, and they're sometimes described as natural antibiotics. So interferon is a family of proteins that's produced rapidly by many different cells in response to a viral infection, and it actually stops the replication of those viruses and keeps it from going into other cells. So it's very important, a very important component as far as blocking those viruses from spreading. Now, because the innate immune response is not able to recognize every single antigen that it's presented with, but it may focus on a few large groups of microorganisms. And so when you kind of look at that large group of microorganisms in general, so it may not be able to focus on a particular one, but some that's in the genetic family makeup, those large groups are called pathogen-associated molecular patterns. Now, the receptors of the innate immune system recognizes these, and then that's actually called a pattern recognition receptor. So that's what we just talked about, was those pattern recognition receptors. So your pattern-associated molecular patterns are actually molecules that are associated with groups of pathogens that are recognized by the cells within that innate immune system, so that once anything is actually presented that's within that family, then the body can actually respond to it. There are 3 groups of pattern recognition receptors. You have secreted PRRs, which bind the PAMPs on the surface of many pathogens, triggering the complement cascade, leading to opsonization and phagocytosis. Phagocytosis receptors bind the pathogen, initiating a signal leading to the release of effector molecules. These are cytokines. Macrophages actually have cell surface receptors that recognize the pathogen-associated molecular patterns as well. And then the third is that you have the toll-like receptors that identify the nature of the pathogen and then turn on the effector response that's actually appropriate for that particular pathogen. And so this signaling cascades leads to the exposure of various cytokine genes. So you have a couple of different ways that this can happen. And moving on to the third line of defense, this is our adaptive immunity. So if the microorganisms actually reach the point where they overwhelm the body's natural or innate resistance, then this third line of defense will come into play. Now, acquired or adaptive immunity is more recently an evolved mechanism that allows the body to actually recognize, remember, and respond to specific stimuli or antigens. Adaptive immunity can result in the elimination of microorganisms and recovery from disease, and the host will usually acquire some sort of specific immunological memory to that particular condition. This is what we refer to as acquired resistance. So this is when the memory cells, or there's some sort of recall that allows the host to respond more effectively if some sort of reinfection with the same microorganism actually happens or takes place. Now, with adaptive immunity, is composed of cellular and humoral components as well. With your humoral, you've got your antibodies and your cytokines. With your cellular, you have your T lymphocytes, B lymphocytes, and plasma cells. Humoral versus cellular mediated immunity. So within humoral mediated immunity, we have what we call antibody mediated immunity. So this is when specific antibodies are actually formed in response to some sort of antigenic stimulation that can be that's actually available to protect the body against any sort of foreign substance. So we have what we call within this umbrella active immunity and passive immunity. Now with active immunity, this is when that antibody-mediated immunity is actually due to some sort of natural exposure or through some sort of intentional injection of an antigen. So, for example, chickenpox. So years ago, most of us developed immunity to chickenpox just through a natural exposure. So getting chickenpox as a child and then developing that rash and, having our body's natural response to that particular virus. But now there's a vaccine that has been developed, so individuals nowadays get their immunity through intentional injections. Now, with passive immunity, this is when you actually achieve some sort of immunity through some sort of exchange or infusion of those particular antibodies or the lymphocytes that will actively make the response for those particular antibodies. So what you can have is you can actually have an artificial passive immunity. This is where you actually are giving a dose of those antibodies to actually protect against that. And then you have a natural type of passive immunity. And so this is where the mom actually passes her antibodies on to the baby in utero. And then once that baby is born, then that baby actually comes with its own set of natural antibodies or a natural form of vaccination against all of the same things that mom has built immunity against, whether it was an active immunity from a natural exposure or whether it was through an immunity through vaccinations. Mom has those antibodies that she passes on to baby, and so baby will maintain those antibodies for the first several months of life. Moving over to the cellular mediated immunity. So this is actually moderated by the link between the T lymphocytes and the phagocytic cells, or your monocyte macrophages. So your lymphocytes are the unique bearers of the immunological specificity, which depends on their particular antigen receptors. So T lymphocytes will actually respond to antigens that are presented by other cells in the context of the major histocompatibility complex proteins. And so these are actually a group of genes that code for proteins that are found on cellular surfaces that will assist the immune system in recognizing those as foreign substances. So in humans, this complex is also called the human leukocyte antigen, or the HLA system. Now, there are several other things that kind of fall up under the cell mediated, and these are going to include things like the T lymphocytes, not necessarily being able to recognize the genetic makeup of allografts, or this is going to be like tissue that's genetically different from a genetically different member of the same species. So this would be something like a organ transplant, like of a human kidney. Also involved will be antigen-presenting cells, cytokines, and interleukins, which we'll discuss all of these in further detail as we kind of progress through the course. So here we have a nice picture that kind of depicts some of the differences between humoral immunity and cell-mediated immunity. Now with humoral immunity, B lymphocytes secrete antibodies that will actually eliminate extracellular microbes, whereas in cell-mediated immunity, you have different types of T lymphocytes that recruit and activate phagocytes to destroy ingested microbes and to actually kill that infected cell. So you can kind of see that process where in each of the different cell lines or different immunities, you actually have an introduction of some kind of foreign element. Then you have some sort of responding lymphocyte, and then from there you have some sort of effector mechanism that takes place. And then from there, the function of that, whether it's blocking the infection and eliminating it through different methods. It could be through phagocytosis or whatnot. But this just kind of lays out a visual depiction of the differences between the different forms. Here we have a nice little table that just kind of gives you another visual of the difference between innate immunity and adaptive immunity, and just kind of comparing where with your innate, where you kind of have that first line of defense with that skin and mucosal epithelials, versus the adaptive, where you have the lymphocytes that kind of initiate that response. And the cells, whereas the innate immunity uses more of those macrophages, neutrophils, natural killer cells, versus the adaptive immunity, where it's primarily going to involve the lymphocytes. So now let's talk about some of the various cells within the immune system. So the cells of the immune system are located within the peripheral blood and different tissues throughout the body. So these cells function differently within the host defense system. So most of these cells are actually derived from bone marrow precursors that are circulating the peripheral blood, which are actually our leukocytes or our white blood cells. So we actually have other immunological active cells that reside in tissue. Now, as we've previously talked about, some of these cells function mainly in the innate immunity while others function in the adaptive immunity, and some of them will actually function in both innate and adaptive immune responses. Now, leukocytes can be functionally divided into general categories of granulocytes, monocytic macrophages, lymphocytes, plasma cells. The primary phagocytic cells are the polymorphonuclear neutrophils, often referred to as PMNs or leukocytes, and then the mononuclear monocyte macrophages. The response of the body to the pathogens actually involves a crosstalk among these immune cells, which is going to include the macrophages, dendritic cells, and the T lymphocytes. So they all actually can send out signals and kind of talk to each other. Now, the role of the innate system within some sort of tissue injury actually is going to involve most particularly the neutrophils and the macrophages, in which these cells are recruited to that site of that injury. And then the macrophages actually have a complex program in which they will release pro-inflammatory mediators that will fight off the pathogen, but they will actually initiate various different systems that will help to clear out any sort of dead tissue and start that tissue repair. Now, the lymphocytes, on the other hand, will actually participate in the adaptive body defenses, primarily through the recognition of a foreign antigen and then producing the antibodies. The plasma cells are antibody synthesizing cells. Now, as you may recall from hematology, you have various white blood cells or various other cells throughout the body that are responsible for various things. Now, as far as related to the immune response, we're going to talk a little bit about some of these. Now, with your granulocytes, this is going to include your neutrophils, eosinophils, your basophils, and then from there, each of those has their own specific role that they play within our immune system. So our neutrophils provide an effective host defense against bacterial and fungal infections. And so what they will actually do is they'll kind of seek out those organisms or those whatever is being exposed into the body, and each of those polymorphonuclear neutrophils can actually start that phagocytosis process within. Now, mature neutrophils are actually found in two of the divided pools, the circulating and the marginating pool. Now, the marginating granulocytes will actually adhere to the vascular endothelium, so along that cell wall of the vessel. And then in the peripheral blood, these cells are only in a transition to their potential site of action. So they're actually just kind of circulating around to get to where they got to go. They're not necessarily, they're just basically circulating through if they're needed. Movement of granulocytes from the circulating pool to the peripheral tissue actually happens through that vessel wall. So once the peripheral tissue once they're actually in that peripheral tissue, those neutrophils are able to carry out their function of actually capturing and destroying any sort of invading pathogen or foreign particle. Now, your eosinophils are also capable of phagocytosis. However, eos and basos don't have as much of that phagocytosis activity that we see with our neutrophils. So basically with our eosinophils, they are a regulator of inflammation. So they do actually have the ability to kill some parasites. So as we learn from hematology, you're sometimes going to see these within some sort of inflammatory response as well. So we can see these with allergic reactions, drug reactions, skin disorders, parasitic infections, collagen vascular diseases, Hodgkin's disease, myeloproliferative diseases, So you can see these in a variety of events. Now, basophils also have a high concentration of heparin and histamine in their granules. So if you have events that are triggered by some sort of antigens like pollens, foods, drugs, insect venoms, then you can get a hypersensitivity type of reaction with those basophils responding as well. We also have something we call mast cells. So what these are actually tissue basophils, and mast cells are bone marrow derived cells that can be found in the skin and mucosal membranes. And so they look a lot like your basophils, except they have a whole lot more of those cytoplasmic granules. So you can see some images here at the bottom. That first one is just a regular neutrophil, a segmented neutrophil. Our second one, you have those larger granules. So you can see how you've got those larger granules, those pink granules, make that eosinophil stand out a little bit different than that neutrophil. Then next we have our basophil, and just a normal basophil, so you can see those deep, large basophilic granules that are inside of that cell. And then our last one, of course, is our mast cell, and you can see where those granules have like completely covered that whole entire cell. So with our mast cells, we're usually going to look for those in some sort of tissue. They're typically not going to be seen within the peripheral blood. So this could be something that's done. Some of the older tests I think of when I think of looking for mast cells is when they were looking for allergic responses in patients that had like allergic rhinitis, or basically doing a nasal swab and looking at that under the microscope to see if you've got those mast cells that are triggering part of that nasal response versus trying to determine if it's something else causing the issue. The mononuclear phagocytic system. So with this system, it's going to have a lot of different functions, including phagocytosis, that has been mediated by receptors in a major fragment of complement. Now we'll talk about complement and that whole activation of that system in another lecture. So the cells within this system originate in the bone marrow from the multipotential stem cell. So with these cells, you're going to see this particular pathway, depending on the microenvironment and the chemical regulators. You're going to see maturation and differentiation of these cells that will happen in various directions. So you have the circulating monocytes that continue to be multipotential, but they can also give rise to different types of macrophages. So again, it's another system that's kind of dependent on what the need of. Now within this system, you're going to have monocytes, the macrophages, and the dendritic cells. Your monocytes you're going to see more within your peripheral blood, and they are able to phagocytize anything that may come within its path. Sometimes you can even see on the microscope where they may just for some reason decide they want to eat a red blood cell for the day. Then you have your macrophages. So here at the bottom you can see a picture of your monocyte in this first image. Here you've got your mono. Not doing very good at this. Beside it, you actually have your macrophage. You can see where it has kind of engulfed a bunch of stuff. It may even be trying to engulf that cell right there, as well as this one. It's already got something in it it's been kind of already ate, so you can see some little granules. It could be bacteria. It could be just the granules from whatever cell it has eaten. And so your macrophages are responsible for the detection and elimination of foreign entities. They facilitate tissue repair and remodeling, resolution of inflammation, maintenance of hemostasis, and you can see them within disease progression. So with these cells, you're going to kind of see them outside of the circulating system, although you can sometimes see them in the peripheral blood. They're going to more or less kind of respond to your tissue sites. This last one, we have the dendritic cells. So they respond to microbes by producing soluble substances called cytokines. They act as messengers between the innate and the adaptive immune system. They are also known as accessory cells, and they actually resemble the microscopic appearance of a nerve cell. So that's where it gets its name from, the dendrites. And so these are called dendritic cells. So they have these long string-like extensions to them. So they actually act as sentinels in tissue that respond to microbes by producing soluble substances, again, those cytokines, and then they act as messengers between that innate and adaptive immune system. From there, their main function is going to be to process antigens and present them on the cell surface of the T lymphocyte of the immune system. Other antigen-presenting cells, like your macrophages and your B lymphocytes, present antigens to differentiated effector T lymphocytes and various immune responses. Here we have a nice table that kind of compares the characteristics of the neutrophils versus the macrophages. So it kind of helps to break down what each of their roles are and how they differ from one another, as well as how much they are alike. Now again, that specific cellular component of the immune system actually is organized around two classes of cells, and that's going to be the T lymphocytes and the B lymphocytes. Now lymphocytes are responsible for recognizing foreign antigens, directly destroying some cells, and being able to produce antibodies as plasma cells. And so they can further be divided down into T lymphocytes, B lymphocytes, virgin or naive lymph cells or lymphocytes that are basically mature T and B lymphocytes that have not encountered or been stimulated by any sort of foreign antigen. So basically they've not been really stimulated at this point to behave any differently. So these naive and memory lymphocytes are both called resting lymphocytes because they have not actively divided, or they're not really performing any of those immune functions yet. Now, there's also been a newly discovered lymphocyte, the innate lymphoid cells. So this straddles the innate and adaptive immune systems with this particular cell. Unlike lymphocytes associated with the adaptive immune system, the innate lymphoid cells cannot perform gene rearrangement of their antigen receptors. They have been implicated in a variety of diseases, especially ones that are associated with the mucosal surfaces, such as the skin, the lungs, the gut. Also within the lymphocytic variations or the lymphocytic line of the immune response, we include plasma cells and natural killer lymphocytes. Now again, immunological development for a mammal. The precursor of the lymphocyte actually arises from the progenitor cells within the yolk sac in the liver and embryo. Then within that fetal development throughout life, the bone marrow will start to become the sole provider of those progenitor cells. And then from there, they will develop into the lymphoblasts. And then there's continued cell development, proliferation of those lymphoid precursors that occur as cells that travel to the primary and secondary lymphoid tissue. Now, mature lymphocytes differentiate into the main categories of either those T or those B lymphocytes, where those T cells arise in the thalamus from fetal liver and bone marrow precursors that will actually go and seed in the thalamus during the embryonic development. And then those B lymphocytes are mostly mature in the bone marrow, and then they function primarily in antibody production or the formation of antibodies. Now, kind of going back to our hematology lessons on lymphocytes, because a lot of students get lymphocytes confused with other cells, and then the lymphocytes can have such a different appearance to them depending on, which particular one they are. But after there's any sort of antigenic stimulation, small lymphocytes can actually start to undergo a transformation. So they start off with those smaller, typical, what we think a lymphocyte to be, and then as they're stimulated, they get a little bit bigger. So when they have that stimulation, when you're looking at them on a right smear, they'll have a relatively larger amount with a deeper blue cytoplasm. So as they're stimulated, you see that cytoplasm get a little bit bigger. You start to see that basophilic color come in a little bit more as those cells are getting a little bit larger. and then you can start to see some of those little granules start to develop within the lymphocyte. Now, the large nucleus has a reticular appearance with a uniform chromatin and prominent nucleoli. So they've had assigned various names to these cells over the years. Again, they may be referred to as reactive, atypical, variant, reticular lymphocytes. But that's basically why they're starting to look like that, because they have had some sort of antigenic stimuli, and it's causing that cell to respond to it. So they're going to start looking a little different. So this is why we see that response to, like in mono, where we get that association with the reactive lymphs. So there has been some sort of stimuli. That Epstein-Barr virus has been introduced into the patient. The patient is responding. Those T lymphocytes are actually responding. So you start to see those lymphocytes start to have that different appearance, and you'll get that reactive response to those cells, and you'll get that weird, what we call reactive lymph, that is typical of when a patient has a monotype infection. And here we have some great images that kind of show you the location of the thymus, as well as the stages of maturation for the T and the B lymphocytes, as far as where you find them, their anatomic location, as far as where the stem cell is produced, versus if it's just an immature cell. You've got those T lymphocytes, that T, associate that with thymus, and then B lymphocytes, bone marrow, as far as where they're located. So, if it's in the bone marrow, it's going to mature and be found in the peripheral blood. So as mature cells, you're going to find them in the peripheral blood for both of those. Plasma cells. So these are not normally found circulating within the peripheral blood, although I wouldn't say that you would never see them, because it is possible. But plasma cells arise at the end of the B cell differentiation into a large activated plasma cell. So these are those large lymphocytes that have that eccentric nucleus, where that nucleus is kind of sitting over to the side. They function in the synthesis of immunoglobulins. They may be seen in the peripheral blood of patients with measles, chickenpox, scarlet fever, or in malignant conditions like multiple myeloma and plasmacytic leukemia. Your natural killer lymphocytes are a very unique cell that has a role both in infectious disease defense and tumor surveillance. They lack the conventional antigen receptors of T and B cells. They destroy target cells through an external non-phagocytic mechanism referred as a cytotoxic reaction. When I say a target cell, I'm not talking about an actual red blood cell target cell, just a cell that the natural killer cell is actually targeting to attack and destroy. These cells are actually just appear as really large lymphs with some granules in it. So again, it's just a lymphocyte. You're not necessarily going to call it a target cell, a natural killer cell. And then there's also a newly discovered lymphocyte, that innate lymphoid cell that straddles the innate and adaptive immune systems. It produces cytokines, as well as performs similar function to the CD4 positive and CD8 positive effector cells. They do not express those T cell receptors. Also want to mention that we have primary and secondary lymphoid organs, so both the bone marrow and the fetal or fetal liver, if it's the fetus, and the thymus are classified as primary or central lymphoid organs. So your bone marrow and your thymus, so they're your primary lymphoid organs. The bone marrow is the source of the progenitor cells. So in mammals, the bone marrow supports the eventual differentiation of the mature T and B lymphocytes, probably from a common lymphoid cell progenitor cell. The bone marrow and the gut-associated lymphoid tissue may function in the differentiation of the progenitor cells into the B cells. So I'm not expecting you to know a lot of detail of how that actually happens. Just know that your bone marrow and as far as in your fetus, your fetal liver tissue and the thymus are going to be your primary lymphoid organs. The thymus exercises control over the entire immune system. So the progenitor cells that migrate to the thymus will actually proliferate and differentiate under the influence of the humoral factor thymosin, acquiring surface marker membrane antigens forming the thymocytes. Now, your secondary lymphoid tissue is going to include the lymph nodes, the spleens, the gut-associated lymphoid tissue, the thoracic duct, bronchius-associated lymphoid tissue, skin-associated lymphoid tissue, and the blood.